Hey scientists, so today we're going to talk about how genes and the DNA are passed on throughout the process. So here we go. All right, so let's start with how do cells divide. Now this is going to be a little bit of review and a little bit of new material. So, mitosis and meiosis are sort of key players in this whole drama. So I'm going to go over that a little bit right now. So how do cells divide? I'm glad you asked. Mitosis, exclamation point. All cells replicate and they divide through mitosis. So this is how we grow and replace aging cells. So as they die, we make new ones. This involves all parts of the cell. So the DNA is the collection of recipes, as you remember from the last video, that codes for life, and that DNA is in every cell, along with the organelles and all those individualized cellular functions. So when a cell replicates and divides, the DNA replicates and divides as well, along with all the other organelles. All right, so how exactly does DNA replicate? The two strands of DNA must be separated in order to be copied. One protein separates the two strands of DNA. Now remember, I said that proteins do a lot of things, from like lovely hair to skin to whatever, digestion, the amylase. One protein sort of unzips the two strands of DNA, and then another protein brings the correct new base pair, remember AT and CG, to pair with the existing base, thus using sort of that as one of the templates. Now remember, A with T, G with C, just sort of when you make the amino acids. All right, DNA replication. So in this way, what happens is two DNA strands are formed from the one. So you've got each new DNA double helix has an old strand. This is called the parent strand, and then a new strand which connects to it, which is the exact sort of the parent strand is the template, and then the daughter strand connects to it. So the original strand is the parent strand. The new strand is called the daughter strand. All right, your DNA. This is crazy. So all the DNA in a cell of a human is called the human genome. We've discussed the Human Genome Project, where they've sort of mapped humanity,'s mankind's, and womankind's, humanity's DNA. So anyway, the human genome has over 3.2 billion base pairs. So this is the crazy part. So if you were to string out one cell's DNA, it would be over 6 feet long inside one single cell. Now, you may wonder, how can 6 feet of DNA fit into the nucleus of the cell? And that is in every cell of your body. It condenses. So the DNA wraps tightly around a spool, just kind of like kite string. And this is called a chromosome. Condensing a strand of DNA is kind of like taking a really long string and then sewing it into a shirt and then balling up the shirt into a very tight wad. So that original string is the DNA. So here's a picture of the original DNA, condensed DNA, and then what it does is condenses into the chromosome. All right, so how DNA condenses? It condenses, like I said, it condenses into, so you've got the DNA, and then the DNA wraps around the proteins. That's kind of like the kite string around the spool. Then you've got the spool DNA, and then that condenses even further into chromosomes. All right, so all the chromosomes can be seen on a karyotype. This is sort of what makes us us at our very genetic level. So karyotypes are made using the amniotic fluid from a pregnant female. So karyotypes, they show the number of chromosomes, the sex of the individual, and they can also show large errors in chromosome structure. So like for instance, we'll get into this a little bit later, but like Down syndrome, where you have 3 chromosomes at 21. And as you can see here, here's a typical male chromosome. You'll notice the XY, and a typical male karyotype, you'll notice the XY, and a typical female karyotype, where you have 2 X's to determine the sex of the offspring. So once DNA replicates, how do cells replicate and divide from there? So the body grows by one cell dividing into 2, 2 dividing into 4, and so on. Skin cells, they live for many days. Blood cells, which carry the oxygen, they can live for months, and our nerve cells, they live forever. So cells divide by the process of mitosis. Remember, this is asexual. So cell with a single copy of DNA. DNA replicates to form chromosomes, which is 2 copies of the DNA. The chromosomes line up in the middle of the cell. Then the chromosomes split half of each chromosome travels to either end of the cell, and then the cell divides to form two new cells with their own DNA. And I know you guys know the interphase and prophase and all that fancy individual stages. All right, so how do parents pass on genes to their children? So the traits are passed on from both of your parents. You have some traits from your mom and some from your dad. So because the traits are coded, they're coded by the DNA, you have some of your mom's DNA and some of your dad's. But the DNA is unique to you from the way that it comes together. No one else has exactly the same DNA as you, unless you're a twin. And then it gets really interesting. All right, so let's do some math. Yay, math! Humans have 46 chromosomes in each cell. If you get 46 chromosomes from each parent, you'll have 92 total. But humans, to be healthy and sort of genetically sort of where we need to be, we can only have 46. So how do we get the 46 chromosomes? By getting DNA from two people. So in the process of meiosis, a cell with 46 chromosomes replicates and divides, but makes the cells with 23 in each individual cell. So cells with 23 chromosomes, they're called sex cells. They're only in certain parts of the body. They're not somatic cells. They're not body cells. They're sex cells. And both males and females have sex cells. All right. Meiosis. Knew where we were headed on this one. Here you've got the original cell with 46 chromosomes. Some DNA can detach from one chromosome and switch with DNA from the chromosome that holds similar genes. This is called crossing over. So when they come over, you've got that synapsis and crossing over. After two cell divisions, 4 cells with 23 chromosomes are formed from there. So you have just the right amount of chromosomes when they come back together, 46. All right, so how do sex cells form new people, new individuals? The sex cells in females are eggs. The sex cells in males are sperm. One sex cell from the mother, an egg, merges from one sex cell with the father, a sperm, and when they come together, you'll remember they're called a gamete. This cell continues to divide until it forms a complete organism. So again, that everything that we are comes from two cells. Mind-blowing. So the individual then, now that the meiosis is done, the individual continues to grow through mitosis. 2 become 4, 4 become 8, da, 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 da. All right, mitosis versus meiosis. Again, just to kind of review some of the differences. So mitosis is 1 division, meiosis is 2. Mitosis forms 2 cells, meiosis is 4. The newly formed cells in mitosis have 46 chromosomes. In meiosis, they have 23. Mitosis forms normal cells. Meiosis is, or normal cells, somatic cells. Meiosis only forms sex cells. Mitosis results in growth, and meiosis results in reproduction, procreation. And that is it. How genes and DNA are passed on. See you next time.